How's it going today, everybody? Today we're going to start looking at our chapter on DNA. This PowerPoint should be findable in the classroom. I'm mostly going to follow along with it, although there are a couple of slides that will be skipped, mostly because the animations aren't going to work on here. So throughout this chapter, we're going to look at DNA as the genetic material. We'll look at replication of DNA. We'll look at DNA, RNA, and proteins and their relationships to each other and the way they function. And then we will also look at gene regulation and mutation. And to start off today, we are going to look at section 1, DNA as the genetic material. So in order to get started with DNA as a genetic material, it's important to understand the context for all of the experiments that led to the discovery or the recognizing of DNA as the main source of genetic material. Before 1928, when Griffith performed his experiments, it was thought that protein was the main source of genetic material, the main things that pass down genes from one organism to another. Griffith's experiments, however, provided evidence that DNA was actually the main source of genetic material. And it did this by looking at two different strains of a bacteria and mice. So this experiment used two strains of bacteria, the S, which is the smooth strain, and that was disease causing. It wasn't able to be fought off by the host mouse's immune system. So injecting a mouse with just a smooth strain of the bacteria, as in section A right here, would cause the mouse to die. There was also the R strain, which is the rough strain, and this one is able to be fought off by the host mouse's immune system. So if you inject just the R strain here, the mouse will live. So Griffith's actual experiment started in C and D, and what he actually did was he heated up the S strain of bacteria in order to kill that bacteria. So when you heat up a cell to a high enough temperature, it's usually going to lyse. It's going to break the cell, killing it, making it non-functional. And as we can see in C, after he injected the smooth strain of the bacteria that had been heated up so that all the bacteria died, the mouse would actually live. It would not get that infection. It would be perfectly fine. But he did notice that when he injected the heated up and killed version of the S strain here, but also some living R strain bacteria, when those were injected together into the mouse, you actually would get a reaction. So even though up in B, when we inserted the rough strain of the bacteria into the mouse, it would live. And when we inserted the S strain of the bacteria into the mouse, it would live as long as the S strain had been killed before. Here in D, when we do both the killed S strain and the R strain, the mouse still dies. And this led to the idea of this principle of bacterial transformation. And the idea behind this was that something in the dead cells of the S strain could cause the R strain to actually transform into the living S strain. And this was confirmed because they could actually take living S strains of the bacteria out of the mouse after this experiment was over. Now, this actually led to a little bit of debate in the scientific community about whether or not DNA was responsible, or if there were some proteins that managed to live through the heating, or maybe some of the RNA or whatever other genetic material was present might have survived, and if there was just a little bit of an amount of that stuff, it could potentially cause that transformation to happen. 16 years later, in 1944, Avery came along, Oswald Avery, and with some better technology, some refined processes, and a little bit better ability to actually look into the molecules that were present, Avery was able to perform this experiment again using a little bit more detailed stuff to show that it was definitely DNA. And so he identified the molecule as DNA, which transformed the R strain of bacteria into the S strain. When the S cells were killed, there was DNA released, and the R bacteria could incorporate that DNA into their cells and change themselves into the smooth version of the bacteria. The way that Avery was able to confirm his results was by treating the killed version of the S strain with a few different enzymes that broke down different options for genetic material. So if he treated it with trypsin, chymotrypsin, or ribonuclease, which are different enzymes that'll break down proteins and RNA, the R strains would still be transformed into the S strains. They would still kill the mice, so transformation would still occur. But if he were to treat it with an enzyme that broke down DNA, that transformation no longer happened. The host mouse would live, and you would not see any bacterial transformation. Now, even with these experimental results, Avery's conclusion was still not necessarily entirely accepted by the scientific community for a while, until Hershey and Chase came along. And at this point, we had even fancier techniques for identifying biomolecules. Specifically, they used radioactive labeling. Radioactive labeling can be used to give DNA or proteins a radioactive signature, which can be identified later. In this case, Hershey and Chase applied two different radioactive labels to a virus they then injected into E. coli cells. For one group, they labeled the DNA with 32P, and for the other, they labeled the proteins with 35S. After labeling, the viruses were mixed with bacteria cells, which they infected before being removed. The bacteria and viruses were then separated and analyzed for the radioactive labels. The test tubes with the 32P labeled DNA only had bacteria cells that were radioactive, meaning that the radioactive labels had been transferred, along with the viral DNA, from the viruses to the bacteria. In the other test tubes, the ones with the 35S labeled viral proteins, radioactivity was only found in the liquid containing the viruses and not in the bacteria. This meant that the proteins were not transferred from the virus to the bacteria. This showed that DNA, which was injected from the virus into the bacteria, was what provided the genetic information needed to produce more viruses. This slide provides a summary of all of the different data from that previous experiment. Go ahead, take a moment, pause the video, and see if you can match these boxes to their corresponding spots on the chart. So, in the group 1 liquid with viruses, we had no viral replication, and in the group 2 infected bacteria, we had no labeled viral proteins, and the new viruses did not have a label. Now let's move on to the structure of DNA. DNA stands for deoxyribonucleic acid, and is composed of long strands of nucleotides. The nucleotides are the building blocks of DNA, and are themselves composed of three major components: a 5-carbon sugar, which is deoxyribose, a negatively charged phosphate group, and a nitrogenous base. DNA consists of two strands of nucleotides coiled around each other to form a double helix, but we'll talk more about that later. The sugar and phosphate groups link to one another to form the backbone of the DNA strand, while the nitrogenous bases bond to each other to hold the two strands together. The 4 nitrogenous bases found in DNA are adenine, guanine, cytosine, and thymine. Often adenine will be represented with just A, guanine with G, cytosine with C, and thymine with T. In RNA, thymine will get replaced with uracil, which will be often represented with just a U. These are divided into two subgroups, purines and pyrimidines. As you can see, the purines have this sort of two-ring structure, while the pyrimidines have just a single ring. Purines will always bond to pyrimidines, which we'll explore on the next slide. Discovering the structure of DNA was a difficult task. DNA is relatively small and difficult to see in much detail in a microscope, and it spends a lot of time coiled in states that make viewing it extra difficult. The first major breakthrough in uncovering the structure was made by Erwin Chargaff. By doing a molecular analysis on DNA samples from multiple species, Chargaff discovered that there were similar amounts of specific base pairs within the DNA he analyzed. Specifically, he found that the amount of adenine always corresponded closely with the amount of thymine, and the amount of cytosine closely matched that of guanine. Using this data, he proposed a rule that became known as Chargaff's rule, which states that cytosine will always pair with guanine and adenine will always pair with thymine. The next breakthrough in determining the structure of DNA came from X-ray diffraction technology. X-ray diffraction allowed scientists to see the molecular structure of DNA far clearer than it had ever been before, and pictures taken by Rosalind Franklin were crucial in determining the double helix structure. A double helix refers to the shape of DNA, often compared to a twisted ladder, which is composed of two nucleotide strands twisted around each other. With the help of X-ray diffraction, James Watson and Francis Crick put together a model of the double helix structure of DNA that conformed with all of the available research on the subject. The two outside strands of the DNA were composed of alternating deoxyribose, which is the 5-carbon sugar, and phosphate groups. The cytosine and guanine bases paired with each other by forming 3 hydrogen bonds, while the adenine and thymine bases paired with each other by forming 2 hydrogen bonds. The double helix structure of DNA is often compared to a twisted ladder. The alternating deoxyribose and phosphate backbone make up the rails of the ladder, and the base pairs form the steps to hold the two rails together. The rails, or phosphate deoxyribose backbone, are held together by phosphodiester bonds, while the steps of the ladder, or the base pairs of adenine and thymine, or cytosine and guanine, are held together by hydrogen bonds. This is a model of what a strand of DNA looks like, without the twisting. The PO4s are the phosphate groups, which hold together 2 deoxyribose sugars. The sugars are bonded to one base, which forms a base pair across the ladder, and then the other side repeats. You've got your deoxyribose backbones here and here. The top strand is said to be oriented from 5' to 3', which are named after the carbons on the deoxyribose sugars. The bottom ladder runs in the opposite direction and is said to be oriented 3' to 5'. The two strands are complementary, meaning their bases pair correctly across from each other. In this case, our top strand is A, C, T, G. So the bottom strand has to be T, G, A, C. That way our A is bonded with a T, our adenine bonded with a thymine, our C at the top is bonded with G at the bottom, our cytosine and guanine. Our thymine at the top is bonded with adenine at the bottom, and then our guanine at the top is bonded with cytosine at the bottom. And these orientations will be important when we look at DNA replication in the next video. The last thing we want to look at is how DNA arranges itself within a cell. If you took out all the DNA of one of your cells and stretched it into a single line, it would stretch about 2 meters, which is about 6 feet long. DNA packs itself very tightly into the cell, and it does so by coiling around small proteins called histones. The DNA wrapped around histones forms what is known as a nucleosome. Those nucleosomes then string together and coil to form chromatin fibers, which supercoil to form densely packed chromosomes. These densely packed chromosomes are visible in the metaphase of mitosis. All of this coiling around itself allows the 2 meters of DNA in each cell to be packed into a space of far less than a millimeter. Well, that's it for this video. See you next time.